Wickersham Chair of Excellence in Agricultural Research, and he's the Scientific Director of the Institute of Plant Sciences at Purdue. His research focuses on identifying genes and genetic resources that contribute to improved crop performance in stressful environments. And he's globally recognized for his work with drought tolerance in maize and sorghum, heat tolerance for maize in Asia, and he's pursuing several really novel research approaches to work that he's doing with sorghum and pearl millet. He's a fellow of the American Society of Agronomy and the Crop Science Society of America. And may I add that, as I shared with Diane a little bit earlier, I'm going to put Jim Camerato. Jim, are you here? Is he here today?
No.
We'll brag on him even though he's not present. But he and Jim in our department I call Renaissance men. They're really good with students. They're really good with the research. They've got it all, the whole package. Don't get the big head in that showcase. But this is a rare situation where you have a faculty member that can cover all those bases and do it with excellence, and Mitch is one of those. Okay? So he's going to share with you today some insights that pertain to plant genetics, plant breeding, and kind of where we are now and what's coming down the pipe. If you join me, please, in giving Dr. Mitch Tuinstra a very warm welcome. Mitch,
Got to put my phone down here so I can keep track of the time. I was told don't run over.
Not going up there, so
Don't worry about it. We'll take care of it. So first off, I just wanted to thank you all and the organizing committee for the invitation to come and talk to you. It's always interesting to come out into the community. Most of the time I'm talking to students and scientists. I train lots of PhDs, and the kinds of discussions that we have with students and faculty and folks that work in the industry, oftentimes quite technical. And so today I was thinking a little bit about what I wanted to talk to you about with regards to the kinds of science that I do. And so I pulled out 4 examples of the kinds of research that I do. The title of my talk is Plant Breeding in the Omics Era. So if I were talking to a bunch of modern scientists, faculty at different institutions, they'd all know what that meant. And I was just as I was sitting here, I'm thinking, omics era? I bet hardly anybody even knows what I'm talking about. Omics. So if you go to a conference today, you'll hear all kinds of students and faculty members talking about genomics and phenomics and metabolomics and transcriptomics. And these are all omicses that are different ways of doing science and trying to understand biology. And my job at Purdue is how do we use the tools of basic plant science to advance plant breeding and develop new cultivars. And so that's what I'm going to talk to you about today. One of the more interesting things that's happened in the last 20 years relates to genomics. Advances in genomics, advances in understanding heredity, the genes that we have in our chromosomes. And it started almost 30 or 40 years ago with a project called the Human Genome Project. And we invested billions of dollars in the Human Genome Project. And now people like me working in agriculture are able to benefit from the technical advances that were made at that time. And so genome sequencing is how do you understand genes that are encoded in the DNA. And genome sequencing has come a long ways. Today, I could extract some tissue or some DNA from some tissue of a corn plant, send it to the core facility at Purdue. In a week or so, I would have the whole genome sequence back for about $1,000. Which maybe it sounds like a lot of money until you think about what is the genome sequence of maize, of corn. It's 2.3 billion A, T, C's, and G's. So for $1,000 in a week's time, you can get the whole genome sequence of a crop that's important in the state of Indiana. If you went to Africa, you might want to work on sorghum, and sorghum has 730 million A, T's, C's, and G's. And so for $400, you can sequence the whole genome of sorghum. You can understand all of the gene just by paying a little bit of money to support the basic research that you're doing in the laboratory. And you're able to use that money to do very directed and almost engineering-like research in crop improvement. So plant breeding strategy. What do plant breeders do? Plant breeders look for genes that control desired traits, meaning we're looking for variation in plants that result, maybe one variety is more resistant to an insect pest, or to a disease, or it's higher yielding, or it has better quality. In many cases, those traits or those attributes are driven by variation in the genes. When Jim Wilcox was at Purdue, he did a lot of work in genetics, and he probably didn't sequence very many genomes. Did you, Jim? But as recently as a few decades ago, we would study genetics by looking at the traits, like cyst nematode resistance, or quality traits, or adaptation to farmer fields in the state of Indiana. So we're looking for genes that have desired results. We combine the favorable genes by making genetic crosses between different strains of plants. We look at the progeny, find those that have the combinations of genes or traits that we are interested in, and at the end of a long process of testing, we distribute those to farmers, and farmers can grow these new varieties, these new cultivars. In my program, what I'm going to talk to you about today is there's lots of different strategies that people use in plant breeding. One of the ones that Jim Wilcox grew up with when he was at Purdue, it's called conventional plant breeding. And the example of conventional plant breeding that I'm going to talk to you about today is this thing that you see on the screen. It's popcorn. Popcorn is a really important crop in the state of Indiana. It's about a $90 million crop. Who's one of the more famous popcorn breeders ever that we would know? Orville. Orville Redenbacher is a popcorn guy. Within 100 miles of campus, a large fraction of the whole global germplasm resource for popcorn is managed by a handful of popcorn breeding companies. And so I work with popcorn. Popcorn is interesting. It's quite different than dent corn. They're related. They're the same species. Popcorn has this issue that it tends to fall down. We call it lodging. And the reason it tends to fall down is the ear sets are too high. The ears on the plant, instead of being 2 to 3 feet high, like in dent cornfield, it might be 1.5 meters or 3 or 4 feet high. It could be eyeball height. So it's very high in the canopy, and that tends to cause problems with standability. We know from a lot of work that's happened over the 20th century that one way to address that is by changing plant architecture. So here's an example in corn. Oh, it's not just show up on the screen. In corn, we can change the plant architecture by using genes. And so we have a number of genes that have been used in the past, and I know some of you actually know John Extell, Dr. Patterson, Dr. Kabisa Ijeda. They work in this space. They're famous plant breeders who've been involved in global food security. A lot of their work is about changing plant architecture to improve productivity. And so what we've done is we've got a gene that we specifically identified that reduces the bottom half of the plant and keeps the rest of the plant the same. So in a normal popcorn plant, you look at that red arrow, the ear height might be about 1.4 or 1.5 meters, so 4 to 5, maybe 6 feet. And by introducing this particular gene called BR2 or Burkitic 2, we can reduce the ear height on the plant. And the result is these plants are more productive at higher populations and they don't fall down. You can actually select for plants that have better tolerance to adverse conditions, and in fact you can reduce the ear height by almost 0.5 meter, 49 centimeters. And we're doing a lot of work with the plant breeding companies in the state of Indiana, popularizing these new popcorn hybrids that we call semi-dwarfs using this modification. Another strategy in plant breeding is called mutation breeding. Can you create variation by mutagenesis? And the example that I wanted to talk to you about today is in a different crop, or a different plant. It's called sorghum. Sorghum is originated in Africa, extremely well adapted to dry conditions, hot temperatures. A lot of people are interested because it's adapted to what people might consider future environments or environments where we're trying to be more sustainable. One of the challenges of sorghum, it's grown both as a grain crop and as a forage crop. It produces a secondary metabolite. It produces a chemical in the cells called durin. So even though it has all these favorable attributes of being very productive in biomass, very good forage, one of the downsides of sorghum is that it produces a secondary metabolite called durin. And if cows eat sorghum, green sorghum leaves, they ingest it into their stomach, the durin breaks down and it's released as cyanide gas. And they call it cyanosis. And so it can be quite dangerous if you're not careful. We know a lot about the pathway that is the biochemical pathway for producing durin. It starts with tyrosine and results in durin that can then be released as cyanide. And what we're interested in, that thing that's crossed out there, that's the first enzyme in the pathway. We want to know, could we break this pathway so that the plants aren't able to produce a secondary metabolite? So we did a mutagenesis study. We looked at 50,000 sorghum plants that had been mutagenized, and we found 2 plants that weren't able to produce durin. So there was no possibility of cyanosis because they didn't produce that secondary metabolite. So now we're using those altered versions of these genes, and we're incorporating those into commercial sorghum hybrids. And these happen to be grain sorghums, forages for hay, and forages for silage. And what we found is that we can produce hybrids that are just as productive as conventional hybrids, but they don't produce any durin. They're completely safe from the standpoint of cyanosis. So farmers that want to produce animal feed that is safe for their animals and be able to take advantage of all of the favorable attributes of sorghum, adapted to drought, adapted to hot weather conditions, adapted to low fertility environments, there is now an option for them to use these kinds of sorghums that were developed at Purdue as a source of genetic materials. And so we're working with the sorghum seed industry to commercialize this new kind of sorghum. And so we call these acyanogenic sorghum hybrids. And we're working with Agronomy Seed Company in Romney. The amazing thing about genomics is I can sequence, I told you a few minutes ago, I can extract some DNA, I can sequence the genome, I can understand the genes that are in any variety. I can now target specific genes for modification. I don't have to do it blindly anymore. It's almost like an engineer saying, if I could just change this one thing, this crop would be a lot better. And so what we're doing is we're something called mutant-based reverse genetics. And we're using this technology in grain sorghum for farmers in Africa. So sorghum has never benefited from work to improve quality the way lots of other crops have. Crops like corn, wheat, barley, potato, those are crops that get lots of investment to improve quality because they're important in Western diets. However, we do the work in those other species that helps us to identify the genes that are controlling those traits. And so what we've done is we've created a population of mutants, 12,000 mutants. We call that a functional genomics platform for sorghum. We've sequenced those, and we know that there's 70,000 different mutations present in that collection. And we put all of that data up on this website at Purdue so that anybody in the world that wants to learn about this is able to log in and look for mutants that they're interested in. And then we deposited the seeds with the USDA so that anybody can request them for their research programs. The question is, how would you use a functional genomics resource to improve a crop like sorghum for African farmers? So if you're interested in quality, one of the quality traits, or one of the attributes of sorghum that's really important is starch. So if you take the grain is about 70% starch. The interesting thing about sorghum starch is that it gelatinizes at a very high temperature, much higher than most other cereals. We don't know why, that's the way it is. And that limits its ability for use in baked goods. So what we did is we said, is there anything that we know about this in sorghum? And there's nothing, almost nothing known. However, we do know that there is a gene in rice called ULK1. So we said, I wonder if sorghum has an ULK1. It turns out that it does. So we went into our collection and we found a mutant, it's called 4308, that has a mutation in ULK1, and asked the question, did we change the gelatinization temperature? And what I show here in this box, and I don't know how easy this is to see from the back of the room, what's happened is we've reduced the gelatinization temperature by changing that one gene. We've reduced the gelatinization temperature of the starch by 13 degrees Celsius. It's huge. It's like these sorghum varieties act like barley in terms of the gelatinization temperature of their starch. Lots of interest in, wow, now we can do things with sorghum that we were never able to do before in terms of producing food products for humans. We're using these mutations and others to make essentially a whole new crop with new quality traits that have never really been recognized in sorghum before. The last area of science that's really popular today is called functional genomics. How do you use functional genomics in plant breeding? And so today I'm going to talk about a project that we're involved with in South Asia. It's called Heat-Tolerant Maize for Asia, HTMA. And so we've developed a collaboration with an international organization called CIMMYT, who has responsibility for maize crop improvement globally. And we are working in South Asia and southern parts of India. It turns out that we're just coming out of what they call their summer season. In the daytime, it's 110 to 115 degrees every day. And at night, it's 80 to 85 degrees. Really oppressive. It's so hot that farmers don't generally grow anything. It's just too hot to grow crops, especially corn. And if I take Indiana corn hybrids, if I go to Pioneer or to Corteva and grow Corteva hybrids in southern India during the summer season, you know what happens? They don't like it very much. In fact, they die before they even mature. And so we've begun a project with CIMMYT looking for tropical varieties, or semi-tropical varieties, that are adapted to these environments. And we're using the tools of functional genomics to develop new cultivars that are adapted to these ridiculously high temperature conditions. And we've been highly successful. This is a large team of scientists. That picture on the top left, I'm one person there. I'm sure you can't even see it. Lee probably can't even see it from here. But what we're doing is we're developing new hybrids that are actually able, nobody thrives at 115 degrees. Even the plant breeders don't thrive at 115 degrees, I can tell you that. But these corn hybrids are able to produce 70 to 80 bushel yields under conditions that are so tough that farmers wouldn't normally grow in those environments. So they're adapted to these hot summer condition environments, and when you plant them in the regular season, every year we get adverse conditions. If you go, everybody's probably wearing, a lot of people I see are wearing sweaters today, so it's hard to think about 115 degree temperatures. But it's not uncommon to have these excessively high temperatures, and we're using the products of this research project to develop heat-tolerant hybrids that are able to grow and thrive in lots of conditions in South Asia. And we've actually just started releasing those hybrids in 2015, and we were just refunded by the USAID program for international development to produce 5,000 metric tons of seed of these hybrids within the next 5 years to start transforming agriculture in these types of stressful environments. The last little comment I have here is about closing thoughts. I talked to you a little bit about a few examples of research that we're involved with at Purdue, developing new forage sorghums, new popcorn hybrids, new grain sorghums with quality traits, new corn hybrids that have these unique adaptation characteristics. Some of the work is important in Indiana, some of it is important across the Corn Belt, some of it is important across the globe. So you can spend a whole career working on projects like this and think you're making some progress and you're making some contribution, but there's still a lot of opportunities for other faculty members like myself, for the students that I train, from South Asia and West Africa, to be trained in the U.S., to work on projects like this, and to ultimately go home where they can have an impact on the agriculture in their own countries and their own regions of the world. What I talked about is about 10 years worth of work at Purdue University and a lot of money from a lot of different people. I'm just on this last slide, I'm just trying to recognize the sources of funding. And that includes people that have been working in my lab and others, support from the USAID for International Development, the Howard Buffett Foundation, the Bill and Melinda Gates Foundation, and from the Department of Energy. Thanks for your attention. I can take some questions if there's any interest.
Where's this going to be in 20 years?
Which one?
Well,
Or ag in general?
Genetics.
Genetics.
Genetic research, that's what you're talking about here.
Yeah, so one of the interesting things today, if you go with, it's amazing actually, there's a new technology that if you read popular press articles, you read about genome editing. Have you ever heard of this? Have people heard about genome editing? It's in People, it's in Time, it's in National Geographic. What I'm talking about are examples of genes, looking at allelic variation or genetic variation and using that variation to create new varieties. With genome editing, we're now able to change the genes in a very specific way, even more like engineering. Change the genes, so change of the genes, for example, of sorghum to look more like corn, corn to look more like sorghum. Really the possibilities are almost endless. And so what we're going to see is going to be, we're going to start seeing the blending of new kinds of crops that have the best attributes of all of the species that are currently available. I'm guessing within, I don't know, at some point in not too distant future, we're going to see C3 plants like soybean. They're going to be able to do photosynthesis like corn. Way more productive, increasing productivity, changing the efficiency of the entire photosynthetic process. We're going to see corn plants that can fix their own nitrogen. So soybeans can fix nitrogen, corn can't. Within a few decades, using some of the modern technology, I don't know how far out in the future this is, but within a few decades, we're going to see some clever young scientist figure out how to manipulate the genes to make corn more productive so that it makes its own fertilizer. We're going to see, so there's lots of examples of things. We know plants can do them already. Some plants can and some plants can't. And we're going to start seeing more and more examples of taking traits or phenotypes of plants like soybeans that can fix nitrogen, and then other plants will be able to do that. Plants that are reasonably efficient at doing photosynthesis like corn and sorghum, supercharged photosynthesis. I think that's what we're going to see in the coming century. Yes?
You sure make it sound easy.
It's so darn easy.
Just one gene, take a gene here, put it in another thing over there, move a gene back over to another one. Nothing to it, just do it.
It is, in some cases. The challenge isn't so much the science today. The challenge is the social implications. So, gene editing, the reason you're reading so much about it is it's really surprisingly easy to do. Today, if you read in the popular press, you'll read about garage scientists. They're doing genome editing experiments in their garage. I read about a guy who wanted to change the way his muscles worked, so he made a genome editing construct and injected it into himself.
Did it work?
I didn't say he was the smartest guy. So, there's tons of social risk, but there's tons of opportunity as well. And the challenge of scientists moving forward, scientists and society moving forward, is looking for that middle ground where science is used in responsible ways to make food production easier, more efficient, so that there's enough food for everybody. Yes?
At the point of history, a lot of this presentation today, a lot of the basis for that came from a former Kiwanian
A
in this club,
Which one?
Bob Pickett.
Oh, Bob Pickett, yes.
Who maintained and brought together a lot of the world's knowledge and actual plant tissue of all the sorghum crops in the world. Do we still maintain sorghum inventory here?
We do. Actually, Gabisa Ejeta manages the mini world collection, we call it, which was founded by Bob, handed over to John Extel, handed over to Gabisa, and now Gabisa is the curator of that small collection.
He's a former member of this club.
Thank you. Thank you for highlighting that. That's interesting. Yes?
It amazes me what you did, what's happened over 10 years, 15 years. Did you ever
put any of Lee's genes in there and find anything bad?
I tried, but they didn't take. Yes.
On a little different note, you mentioned southern India, and there are places in the world that just aren't suitable for growing anything. Where is the biggest challenge in the world that still has some potential for making some progress?
Say that one more time. You want to know the place that has the most potential?
If I look at places like Antarctica,
Yes.
you're probably not growing a whole lot of crops in Antarctica.
Not yet.
You're
Yeah.
beginning to see in southern India that we can in fact make some productive gains in food production. Where are those biggest food production challenges in the world today?
In India, right now, the biggest challenge, that is a huge production area, highly managed, highly educated agricultural force of farmers. The challenge that they have is that in India, it's a monsoonal environment. The monsoons are blowing in as we speak, meaning the rains are just about to begin, or depending on where you are, north or south, they've just begun. It's going to rain steadily for the next few months, and then it's going to stop raining, and depending on where you're at and what the relative conditions are, it's going to stop raining, and then you're going to see it's not going to rain very much, if at all, from late fall until next June.
Wow.
Farmers have known forever those are two growing environments, the rainy season and the dry season. In the rainy season, we grow rice, and in the dry season, we grow sorghum or wheat, depends on where you're at. The challenge for farmers in India is that the monsoons are changing. They're not very predictable anymore. It used to be the place that I'm going to be going to in a couple weeks. On June 15, it's probably going to start raining. And you just plan on it. I'm going to farm according to the calendar. Now, it's starting to rain early. It's starting to rain late. It stopped raining a month or so earlier than it should. It's raining longer than it should. What is the cause of the variability? I don't know exactly. And so a lot of this variability in climate, in environment, is causing challenges for farmers that work in these environments where it's highly dependent on rainfall. Africa's subcontinent is probably one of the highest potential in places where there just hasn't been a lot of technology. There's lots of good agriculture, farm ground, but there's been a lot of challenges with politics and society and other things that have limited ongoing investment. I work in places where we go from very tough, very challenging situation. You go 10 years of improve, and all it takes is a little political mix-up, and all of a sudden you're back to where you started. And it's going to take some sustained investments in agriculture in these different parts of the world and different parts of Africa to see some of the types of systems that we have, highly developed systems that we have in the United States. And you're going to start seeing changes in productivity. So today, some of the places I work, it's not uncommon to produce 10 bushel yields in corn. 10 to 20 bushel yields. It doesn't sound like much, does it? Was I telling you 242 bushels over in Benton County, a 1,000-acre farm? That whole farm, 1 acre, could have produced as much as more than similar-sized farms on 1 acre than what a 240-acre farm or a 1,000-acre farm in Nigeria or Niger or Senegal. So as we start seeing investments, as we start seeing sustained development, and it's getting better, I think, in some of these places, we're going to see greater opportunities for food production, technology, and innovation. Yes?
It sounds like the work you're doing has implications for work outside of this world, and like on Mars.
Mm-hmm.
Have you done any conversations or any work with the potential for agriculture and the manipulation of these genes for work in a different environment like Mars?
One of my colleagues at Purdue works in that space, Carrie Mitchell in horticulture.
Sure, I don't care.
So Carrie does a lot of work in NASA programs, and at the moment it's how do you take Earth-like environments and simulate them in Mars. What is the potential for Mars? I don't know what the potential for Mars is. Everybody's watched The Martian, right? The movie called The Martian. Is it called The Martian?
That's it, yeah.
And it's like he calls himself the greatest botanist on the planet or something like that, the only person on the planet. What is the potential for colonization of other planets? there's some hope for that, but I would much prefer to preserve a world that we have here.
Amen.
I hope we figure out ways to modify the environments of Mars and other places so that there's other places to go and be. But I hope we at the same time don't destroy our own planet. That's because I think it's going to be hard to beat the world, the Earth, as a place when I walk around out in the cornfields and sorghum fields and in the forest behind the house. It's going to be hard to beat.
What is the agronomist saying about the future of corn and soybeans in the state this year?
So some of you may have noticed that it's been raining a lot. And so I was ready to plant on April 25. That was the rule. Guys, we've got to be ready to plant on April 25. May 1, we hadn't planted. May 10, we hadn't planted. May 15, May 20, May 25. And we were ready to plant. I mean, the tractor was practically idling. On June 2, 6 o'clock on a Sunday morning, I got up and I said, here, getting really close to being able to work some ground. I went to see Jim Beatty out at the farm. At 6 o'clock in the morning, he saw me across the farm. He said, Mitch, is that you? He sent me a text. I said, yeah, it's me. He said, come get me. So we drove around the farm. He said, when are we going to start? I said, we got to start now because it's getting late. And so we started working ground on Monday, June 3, and we were 90% done by Wednesday, June 5, just because that was the first chance we had. We worked from 6 o'clock in the morning until 10 o'clock at night, but that's what you do when you have to. What are the prospects this year? It would be helpful if we were able to get some warmer days, so that all of this corn that got planted in early June has a long ways to go. And 60-degree, 65-degree daytime temperatures, they slow things down. We don't need to be slow. We need things to speed up. So it's a little scary, actually. Running out of season this year would be what I would be considering. An early frost.
Saturday and Sunday.
Questions?
I just wanted to make a comment about plant breeders. I was on the sales side of Pioneer for a long time, and I marvel at what you guys do. And I had a farmer tell me one time, when seed corn got to $300 a bag, he said plant breeders are underpaid.
Mm.
Because he had an appreciation for what you guys do.
Thank you. The technology today, the real challenge is, I didn't talk about the other half of the things that I do. So we're doing a lot. So what is Purdue known for? They're known for agriculture, and they're very well known for engineering. And ever since I was a grad student here in the 90s, it's like, we need to figure out how to work with these engineers. There's lots of smart engineers on campus. And so a couple years ago, we started very intently in my job as the scientific director for the Institute of Plant Sciences, walking across campus, sitting down with engineers, getting them excited about agriculture. Turns out that most people, regardless of their background, are worried about food, sustainability, environment. And if you go to them and say, man, there's a global challenge of this, can you help me solve it? They get excited about it. And then you tell an engineer, yeah, you probably can't get it done. You probably can't even do it. You probably can't get that. You probably can't do that. And all of a sudden, you got an energized, different set of skills. So one of the big changes for me personally is we've mobilized a large group of engineering scientists across campus that are now engaged at the agronomy farm. And if you drive around the agronomy farm today, you'll see UAVs or drones with sensors and cameras on them flying around, collecting data. I work with a world-famous solar energy scientist. We're developing entirely new kinds of solar energy arrays that you can grow corn under them, where there's no depression in yields, because they let the power light through, the photosynthetically active radiation through, but they only capture the light that the plants can't use during the growing season. We have lots of examples like that of new people at the farm who don't know beans about agriculture. But they're really smart and they have interesting technology, and it's a good time for agronomists to engage.
You
Yes.
don't believe that, try to start a new tractor.
Yeah. I have a new John Deere, and I have to ask my technician, Andy, could you drive the tractor? I don't even know how to start it. Thank you, yes.
Are you saying that someday the corn and the bean will be the same plant?
I don't think they'll be the same plant because they're fundamentally different biology, but the best attributes of corn will be in soybean plants, and the best attributes of soybeans will be in corn plants. Those will be the easy things. And then the clever biologists are going to come behind and say, we've never thought about that before, but you can take these genes from these microorganisms that grow in hot springs in Yellowstone and put them into corn, and maybe the popcorn pops on the ear. I don't know that that's true. So I'm just kidding. But you're going to have all kinds of clever ideas, people doing things that are unexpected, and some of them will even work. Yeah.
Let's give him a round.
Thanks again for hosting me. It's been a pleasure coming. Hope we get a chance to say hello to a few of you on the way out.